angiosperms and that are obviously complete plants. In approximately 30% of angiosperm species, formation of sperm cells occurs even while pollen is still located within the anther. In the majority of angiosperm species, the pollen is released from the anther at about the time the generative cell has formed, and sperm cells are not produced until after the pollen has been carried to a stigma. After a pollen grain lands on a stigma, it germinates by producing a pollen tube that penetrates into the loose, open tissues of the stigma. The pollen tube absorbs nutrients from the stigma and grows downward through the style toward the ovary. The microgametophyte is protected and nourished by the style tissue. As the pollen tube grows downward, it carries the sperm cells to the ovule. Almost all of the pollen cytoplasm is located at the tip of the pollen tube. The rest of the tube and the pollen grain are filled with a giant vacuole. Pollen grains are too small to store much starch, fat, or protein. If they could not absorb nutrients from the style, the pollen tubes could not grow long enough to reach the ovules. Figure 9:11 pollen grains of Cabe, with the pollen wall forming hexagonal ridges. The numerous holes are germination pores where a pollen tube can emerge after the pollen lands on a stigma. Pollen of Lycium has a single long groove from which the pollen tube emerges. Pollen of Macrolobium has 3 germination grooves. Figure 9:12 in lily, the pistil consists of 3 carpels fused together. This ovary has been cut across to reveal the 3 ovary locules or chambers. In each chamber, 2 ovules are visible. Ovules are attached to the placenta by their funiculus, which contains both xylem and phloem. The central part of the ovule is the nacellus with just 1 megasporocyte, unlike anthers, which have numerous microsporocytes. Magnification of a single ovule. Surrounding the nacellus are the integuments. Growth of the ovule has been by mitosis, but now one cell, the megasporocyte, megaspore mother cell, is undergoing meiosis. Figure 9:13. This is the tip of a growing pollen tube. It was treated to make the nuclei fluoresce so that they can be found easily despite being so small. The lowest nucleus is probably the tube nucleus. The upper 2 are probably sperm cell nuclei, each located within a sperm cell. The 2 sperm cells themselves are located within the protoplasm of the pollen tube. Megagametophyte. Within the ovule, the surviving megaspore develops into a megagametophyte. In one type of development, the nucleus undergoes 3 mitotic divisions, producing 2, 4, and then 8 haploid nuclei all in a single, undivided cell. The technical term for a multinucleate megagametophyte is embryo sac. The nuclei migrate through the cytoplasm, pulled by microtubules, until 3 nuclei lie at each end and 2 in the center. Walls then form around the nuclei, and the large, 8-nucleate megaspore becomes a megagametophyte with 7 cells, one of which is binucleate. The 7 cells are one large central cell with 2 polar nuclei, 3 small antipodal cells, and an egg apparatus consisting of 2 synergids and an egg. Like the microgametophyte, the megagametophyte is a distinct plant. As with the pollen, the megagametophyte obtains all of its nourishment from the parent sporophyte. Plants and people. Box 9-1 Flowers, fruits, seeds, and civilization. Flowers and the fruits and seeds that result from them have always been important to us. One of the first hominids, animals on our line of evolution, was Australopithecus africanus. Fossils of its teeth, 3 million years old, show that it was adapted to eating plants. Homo habilis lived only 1.8 million years ago and likewise had a diet strongly dependent on plants. Our species, Homo sapiens, appeared evolutionarily about 500,000 years ago and survived by hunting game and collecting fruits, seeds, and edible roots. Approximately 11,000 years ago, a momentous change occurred. Small groups of humans began cultivating plants rather than just gathering them. This happened in the Middle East, an area that is now Iran, Iraq, and Syria, also known as the Fertile Crescent. The plants were common, wild species such as wheat, barley, peas, and lentils. Farming required major changes in human society. People had to stay in one place to tend and protect crops rather than follow herds of game animals. Permanent villages were necessary, as were the rules and regulations needed when people lived together in close proximity. Land, huts, and harvests are tempting targets for thieves, and thus, defense, both individual and collective, became a necessity. Government had to be created. Civilization advanced rapidly. Neolithic agricultural societies were widespread by 600 BCE, and the ox-drawn plow was in use as early as 4500 BCE. New plants were cultivated: olives, date palms, and grapes for eating and for wine. Independently, the peoples of Southeast Asia domesticated rice and soybeans. In the New World, the Incas, Mayans, and Aztecs cultivated potatoes, corn, tomatoes, beans, squashes, cocoa, pineapples, and peanuts. Table B9-1 locations where plants were domesticated. Figure B9-1 images like this occur frequently in ancient Sumerian art. The person is either a priest or a king, and the object he faces is a very strange plant. In earlier depictions, the plant is recognizable as a date palm, but through the years this important food tree became a symbol for life itself, and artists began to draw it more stylized and with mystical symbols surrounding it. The person is holding a cone-like object in one hand and a pail in the other. Date palms grow as carplet plants and staminate plants. To ensure a good crop, pollen can be collected in a pail and then applied to the carplet plants with a brush to increase the rate of pollination and fruit production. The scene, carved before 2000 BCE, indicates that the very earliest farming societies understood this sophisticated aspect of botany. The importance of fruits and seeds to the survival of both individual humans and societies is reflected in the prominent position they were given in early art. Ancient Egyptians carved likenesses of date palms, barley, and wheat as long ago as 3000 BCE, and on Crete in 1800 BCE, Minoan artists depicted, in addition to the plants already mentioned, figs, saffron crocus, pomegranate, and lupin. They also showed several plants that must have been grown purely as ornamentals: lilies, myrtle, narcissus, and roses. As societies become wealthier, time and resources become available for leisure and pleasurable pursuits. Gardens dedicated to beautiful, fragrant flowers, not food crops or medicinal plants, arose early. Expeditions in search of exotic ornamental plants are described in the oldest epic poem in the world, the story of Gilgamesh of Sumer, and an ancient Egyptian monument, the Palermo stone, records a plant-gathering expedition by King Snefru in 2900 BCE. Inventories list the extensive plant-collecting trips and gardens of Ramses III. Growing beautiful flowers and enjoying exotic fruits have never lost their popularity. After the Spanish discovered America, the initial exploitation was for gold and silver, but there soon followed expeditions dedicated solely to gathering plants, especially ornamental ones. Two of the many famous plant explorers sent to the United States are David Douglas, who explored the Pacific Northwest for the Horticultural Society of London in the 1820s, and the father and son team, André and François-André Michaux, sent by the government of France in 1785. Even today, gardening and growing flowers for pleasure are considered by many an essential part of our civilization. As technology increases, cultivating flowers, or at least having a potted plant on a windowsill, seems to maintain contact with our past. Figure 9-14. Shortly after landing on a stigma of the correct species, a pollen grain may germinate by pushing out part of the wall as a pollen tube. This penetrates the loose tissues of the stigma and style. This was photographed with ultraviolet light, which causes the pollen wall and collos to fluoresce and shine. Tissues of the style allow the pollen tubes to grow through them easily. Some styles are hollow and lined with a rich, nutritious transmitting tissue. Other styles are solid but also have transmitting tissue. Sunflower style. These pollen tubes have penetrated deep into the style. The bright spots are collos plugs, which seal off the protoplasm at the tip from the empty parts of the pollen tube. Figure 9-15. As a megaspore develops into a megagametophyte, 3 sets of mitosis occur without any cytokinesis. Thus, the megagametophyte is temporarily an 8-nucleate coenocyte. Then nuclei migrate, cytoplasm accumulates around each, and walls are established. Much of the egg is not covered by wall. The central cell is binucleate and is mostly vacuole. Table 9.3 Gametophyte development. Fertilization. Syngamy of sperm and egg involves both plasmogamy, fusion of the protoplasts of the gametes, and karyogamy, fusion of the nuclei. As a pollen tube grows downward through the style toward the ovule, it is guided to the ovule's micropyle by some means. It penetrates the nacellus and reaches the egg apparatus, then enters one synergid. The pollen tube tip bursts and releases both sperm cells, one of which migrates through the synergid protoplasm toward the egg. As it does so, the sperm cell's plasma membrane breaks down, and it loses most of its protoplasm. The sperm nucleus enters the egg, then is drawn to and fuses with the egg nucleus, establishing a diploid zygote nucleus. Because the sperm sheds its protoplasm as it passes through the synergid, it contributes only its nucleus with the set of nuclear genes during karyogamy. The sperm does not carry mitochondria or plastids into the egg, and thus, organellar genes from the pollen parent are rarely inherited by the zygote. Instead, the mitochondrial and plastid genes of the embryo are inherited only from the ovule parent. Conifers are different. Their sperm cells retain their plastids while destroying their mitochondria. The zygote inherits mitochondria and a nucleus from the egg, a nucleus and plastids from the sperm cell. In angiosperms only, the second sperm nucleus released from the pollen tube migrates from the synergid into the central cell. It undergoes karyogamy with both polar nuclei, establishing a large endosperm nucleus that is triploid, containing 3 full sets of genes. Because both sperm nuclei undergo fusions, one with the egg nucleus and the other with the polar nuclei, the process is called double fertilization. The endosperm nucleus is extraordinarily active and begins to divide very rapidly by mitosis, with cell cycles lasting only a few hours. The endosperm nucleus initiates a dynamic cytoplasm and the central cell enlarges enormously, usually without cell division, into a huge cell with hundreds or thousands of nuclei. Finally, nuclear division stops, and dense cytoplasm gathers around the nuclei. Walls are constructed, thus forming cells. An example of this is a coconut full of milk. The hollow center of the coconut is one single cell, and the milk is its protoplasm. The white coconut, meat, is the region where nuclei form cells. A green, immature coconut is full of milk but has almost no meat. As it ripens, the cenocytic milk is converted to a thick layer of cellular meat. All of this tissue, both cenocytic and cellular, is called endosperm, and it nourishes the development of the zygote. No other megagametophyte forms as much endosperm as a coconut does. More typical examples are grains such as wheat, rice, oats, and corn, in which most of the grain is endosperm. Endosperm of these also pass through a milk stage, but by the time of harvest, the endosperm has become cellular, starch-filled, and dry enough to be hard. Figure 9.16. In many species, endosperm development is accompanied by some cytoplasmic division, and the endosperm is therefore a mass of multinucleate cells, each with variable amounts of protoplasm and nuclei and with irregular shapes. This is the only plant tissue in which nuclear and cytoplasmic divisions are so poorly coordinated, possibly because the endosperm is just a temporary tissue that is consumed by the embryo before or soon after seed germination. Embryo and seed development. As the endosperm nucleus proliferates, the zygote also begins to grow, but always by both nuclear and cellular divisions. A coenocytic stage never occurs in the embryo. The zygote grows into a small cluster of cells, part of which later becomes the embryo proper, and the other part becomes a short stalk-like structure, the suspensor, which pushes the embryo deep into the endosperm. The suspensor is usually delicate and ephemeral in angiosperms. It is crushed by the later growth of the embryo and is not easily detectable in a mature seed. Cells at one end of the suspensor continue to divide mitotically, developing into an embryo. The cells at first are arranged as a small sphere, the globular stage. The end of the embryo farther from the suspensor initiates 2 primordia that grow into 2 cotyledons in basal angiosperms and eudicots, such as beans and peanuts. While young, the cotyledon primordia give the embryo a heart shape. This is the heart stage. In monocots such as corn, only one cotyledon primordium grows out. Dicot is an abbreviation of dicotyledon, those plants whose embryos have two cotyledons. Monocots are monocotyledons, plants with only a single cotyledon on their embryos. Conifers like pine have numerous cotyledons. Later, in the torpedo stage, the embryo is an elongate cylinder, a short axis is established, consisting of radical, epicotyl, and hypocotyl. Finally, vascular tissue differentiates within the embryo. The epicotyl may bear a few small leaves, and the radical often contains several primordia for lateral roots in its pericycle. Once mature, the embryo becomes quiescent and partially dehydrates, and the funiculus may break, leaving a small scar, the hilum. In green peas, the two halves of each pea are the two cotyledons, and the stalk attaching each pea to the pod is the funiculus. In most basal angiosperms and eudicots, cotyledons store nutrients used during and after germination. During embryo development, the cotyledons become thick and filled with starch, oil, or protein, whereas the endosperm, which is supplying the nutrients, shrinks. When the seed is mature, the cotyledons are large, and the endosperm may be completely used up. We are eating mostly cotyledons when we eat beans, peas, peanuts, almonds, pecans, and other seeds that easily separate into two halves. Figure 9.17. And in many species, endosperm development is accompanied by some cytoplasmic division, and the endosperm is therefore a mass of multinucleate cells, each with variable amounts of protoplasm and nuclei and with irregular shapes. This is the only plant tissue in which nuclear and cytoplasmic divisions are so poorly coordinated, possibly because the endosperm is just a temporary tissue that is consumed by the embryo before or soon after seed germination. In monocots, the one cotyledon generally does not become thick and full. Instead, the endosperm remains and is present in the mature seed. When eating cereals such as wheat, rice, oats, and corn, we are eating almost purely endosperm. During germination, the cotyledon acts as digestive absorptive tissue, transferring endosperm nutrients to the embryo. Some eudicots are intermediate. Cotyledons store some starch and protein, but a considerable amount of endosperm remains in the seed, and both methods of nutrition are used during germination. A mature seed in which endosperm is rather abundant is an albuminous seed. If endosperm is sparse or absent at maturity, the seed is exalbuminous. Figure 9.18. Embryo development. Globular stage. Heart stage. Torpedo stage. Embryo root, cotyledons, and hypocotyl are present. At this stage, the first xylem and phloem may become distinguishable in the hypocotyl. Mature embryo. A shoot apical meristem is shown. In some species, even some leaf primordia and a small stem are present. Figure 9.19. This bean seed has begun germinating. The radicle has extended and will develop into a taproot. The two cotyledons were digesting their carbohydrates and proteins and transporting nutrients to the root and shoot apical meristems. Beans have a well-developed epicotyl with several partially expanded leaves. One is visible here. These cells of bean cotyledon are full of starch and reserve protein. The small leaves present on the epicotyl are expanding during germination. Because they were rather completely formed before the embryo became dormant, the leaves can now mature rapidly, and photosynthesis begins almost as soon as germination is complete. The hypocotyl is curved. This allows the leaves to be dragged gently up through the soil, protected by the cotyledons. Most of a corn seed is the endosperm. The embryo is less than half the volume of the seed. During germination, the one cotyledon secretes digestive enzymes into the endosperm and absorbs the resulting monomers. Corn seeds also produce many small leaves before becoming dormant, and they can begin photosynthesis immediately after germination. The leaves are protected from the soil by a tubular sheath called a coleoptile. The amount of embryo growth and development that occurs before dormancy sets in is extremely variable. Orchids, bromeliads, and a few other species have small, dust-like seeds in which the embryo is only a small ball of cells with no cotyledons, radicle, or vascular tissue. In seeds of most angiosperm species, all of the parts are present, and the epicotyle contains 2 or 3 young leaves in addition to cotyledons. These leaves can begin photosynthesis immediately after germination. In corn, the embryo is even more advanced. It contains as many as 6 young leaves while in the seed. A fully mature corn plant often has only 10 or 12 leaves. Over half of the leaf production of the new sporophyte occurs while it is embedded in the parental gametophyte, which is itself embedded in the previous sporophyte. The embryo and endosperm develop from the zygote and megagametophyte central cell, respectively, both located in the nacellus of the ovule. Soon after fertilization, or even before, synergids and antipodals break down in most species. The nacellus expands somewhat but later is crushed by the expansion of the embryo and endosperm and is usually not detectable in mature seeds. The integuments that surround the nacellus expand and mature into the seed coat as the rest of the ovule grows. In their last stages of maturation, they may become quite sclerenchymatous and tough. It is seed coat color that lets us distinguish between black, red, and pinto beans. Figure 9.20. This seed coat of bean has an outer layer of sclerids and inner parenchymatous layers. The cotyledon is part of the embryo, and you might expect to see endosperm or nacellus between the cotyledon and seed coat. However, those have been crushed during the later stages of seed development. Fruit development. As the ovule develops into a seed, the ovary matures into a fruit. Development varies with the nature of the carpels as well as the nature of the mature fruit. The stigma and style usually wither away, as do sepals, petals, and stamens, although they may persist at least temporarily. Often 3 layers become distinct during growth. The exocarp is the outer layer, the skin or peel. The middle layer is the mesocarp or flesh, and the innermost layer, endocarp, may be tough like the stones or pit of a cherry, or it may be thin. The relative thickness and fleshiness of these layers vary with fruit type, and often 1 or 2 layers are absent. The entire fruit wall, whether composed of 1, 2, or all 3 layers, is the pericarp. Figure 9.21. In apples, the petals die and fall off after pollination. Stamens and sepals die and dry out, but they remain present, whereas carpels and receptacle develop into a fruit. In most species, sepals, petals, and stamens all abscise after pollination. Figure 9.22. A pecan nut is only the seed and innermost part of a fruit. The exocarp and mesocarp are tough, fibrous, and completely inedible. The fruit opens while on the tree, allowing the endocarp and seed to be shed. Coconuts in stores are usually just the endocarp and seed. In nature, these are surrounded by a thick, fibrous, and buoyant mesocarp and a thin epicarp. A dark spot on the endocarp is a soft spot where the seedling root and shoot will emerge during germination. Before moving on to the next section, think about a peach tree in full bloom. The tree has thousands of flowers, each with stamens and carpels. When it has finished producing pollen and megaspores, it has technically finished its own reproduction and now has tens of thousands of offspring in the form of microgametophytes and megagametophytes. Bees carry pollen from other peach trees to the stigmas of this one, and before long, it has thousands of pollen tubes growing inside its styles. You see one diploid peach tree, but inside it are thousands of haploid pollen tubes and megagametophytes. All of the water, minerals, and photosynthates needed to keep all of these thousands of plants alive are being supplied by the leaves, roots, xylem, and phloem of the diploid peach tree. We cannot say that the haploid plants are parasitizing the tree because they are essential to its production of seeds. After fertilizations have occurred, hundreds of zygotes develop into embryos, each of which is the offspring of microgametophytes and megagametophytes and the grand offspring of the peach tree itself. This tree has other grand offspring as well, produced by its pollen that was carried to other peach trees. This tree is the ovule parent of its own seeds and the pollen parent of thousands of seeds in many other trees. Flower structure and cross-pollination. The production and development of spores, gametes, zygotes, and seeds are complex, elaborate processes, but they are not the only functions of flowers. Flowers are also involved in the effective dispersal of pollen and seeds. Because numerous mechanisms carry out these processes, numerous types of flowers and fruits exist. Cross-pollination. Cross-pollination is the pollination of a carpel by pollen from a different individual. Self-pollination is pollination of a carpel by pollen from the same flower or another flower on the same plant. In any plant population, there is genetic diversity. Random mutations will have produced some new versions of genes that offer improved fitness and some that are deleterious. For example, people with blue eyes have the same genes for eye color as do people with brown eyes. It is just that they have different versions, alleles, of the eye color genes. With cross-pollination, sperm cells and egg cells from different plants unite, resulting in new combinations of genes, at least a few of which may be better adapted than either parent. But self-pollination has about the same result as asexual reproduction because all genes come from the same parent. No possibility exists of bringing in new alleles that might provide more fitness than those inherited from the parent. However, if a plant is isolated by distance or lack of pollinators from potential cross-pollination partners, self-pollination allows it to set seed and propagate its genes rather than lose them when the plant dies. Many mechanisms have evolved that decrease the probability of self-pollination and increase the chances of cross-pollination with its accompanying genetic diversity. Stamen and style maturation times. Self-fertilization in flowers that have both stamens and carpels is prevented if anthers and stigmas mature at different times. In many species, anthers release pollen while stigma tissues are immature and unreceptive. The style may not have elongated yet, and the stigmas may be near the base of the flower while the anthers are at the top, elevated by elongated filaments. Exposed pollen lives only briefly, being susceptible to desiccation in dry air and to damage to its DNA by ultraviolet light. When the stigma and style become mature, there may be no living pollen left in the flower, thus, all pollination is affected by younger flowers just opening their anthers. This is not a very effective means of ensuring cross-pollination. On plants with many flowers that do not open simultaneously, older flowers could be self-pollinated by freshly opened flowers of the same plant. Even if pollen does come from other plants, many of the near neighbors are probably closely related because many of the seeds of a plant fall and germinate near the plant, producing a cluster of plants that are at least as closely related as first cousins. Stigma and pollen incompatibility. In many species, especially many important crop species, self-pollination is inhibited by compatibility barriers, chemical reactions between pollen and carpels that prevent pollen growth. In one incompatibility system, as a pollen tube grows, the stigma and style test proteins on the tube's surface. If one of these proteins is produced by a gene that matches an incompatibility gene in the carpel-bearing plant, the stigma and style block any further growth of the pollen tube. In self-pollination, all pollen tube genes match those of the stigma and style, and blocking occurs. The plant cannot pollinate itself. Figure 9.23. The stamens of this cactus flower are mature and shedding pollen, but the stigmas are pressed together and are unreceptive. Later, the stamens wither and the stigmas spread open, ready to receive pollen from a different flower. In another common system, the critical proteins are deposited on the outer surface of the developing pollen grain by the anthers tapetum. Any match of proteins produced by the incompatibility genes blocks germination of the pollen grain. The interaction between a pollen tube and a style involves a haploid genome and a diploid one, whereas the second system involves two diploid genomes. The diploid-diploid system involves twice as many genes and therefore has a much greater probability of pollen rejection. With the diploid-diploid system, not only is self-pollination prevented, but inbreeding between close relatives is blocked as well. Monoecious and dioecious species. Among incomplete flowers there is a significant difference between flowers that lack sepals or petals and those that lack stamens or carpels. The latter two organs are essential organs because they produce the critically important spores. If either organ is absent, sexual reproduction is dramatically affected. Flowers that lack either or both essential organs are not only incomplete but also imperfect flowers. If a flower has both, it is a perfect flower even though it may lack either sepals or petals or both. Sepals and petals do not produce spores and are considered non-essential organs. Figure 9.24. This pollen grain has started to grow on an incompatible stigma. but the stigma has blocked it, causing kalos to form. The kalos fluoresces in ultraviolet light, so it shines brightly in this micrograph. It is necessary to consider the entire plant and the entire species as well as individual flowers. Stamens produce pollen that results in sperm production, and carpels are involved in egg production. Thus, a species must have both types of organs. Plants that have perfect flowers satisfy this requirement. But if some flowers of the species are imperfect, having no stamens, for instance, then other flowers must have stamens by being either perfect or by being imperfect because of a lack of carpels. A large number of combinations is possible. A species may have individuals that produce only stamenate flowers and others that produce only carplet flowers. This is dioecious, and the species is said to be dioecious. Examples of dioecious species are marijuana, dates, willows, and papaya. In dioecious species, the life cycle actually consists of 4 types of plants: microgametophytes, megagametophytes, stamenate sporophytes, and carplet sporophytes. Monoicy is the condition of having stamenate flowers located on the same plant as carplet flowers. Monoecious species include cattails and corn. Ears are clusters of fertilized carplet flowers, and tassels bear numerous stamenate flowers. In some members of the cucumber family, the type of flower produced varies. Young plants and those growing in a poor environment produce stamenate flowers, whereas older plants and those growing in a good environment produce perfect or carplet flowers. If fertilization occurs, the carpels develop into large fruits, and only a healthy, robust plant can afford to do this. A young or poorly growing plant cannot supply enough carbohydrate and protein for fruit development, but it can supply enough to produce pollen. Dioicy is an extreme adaptation that ensures cross-pollination. A plant that produces only one type of spore cannot pollinate itself. It is similar to the condition of separate genders in mammals. No individual can fertilize itself. Both conditions ensure that fertilization is by sex cells that are not identical genetically, thus increasing the genetic diversity of the offspring. Animal-pollinated flowers. The evolution of animal-mediated pollination had a dramatic impact on the evolution of flowering plants. In wind-pollinated conifers, things such as brightly colored petals, fragrances, and nectar are a waste of material and energy. But once insects began visiting early angiosperms, mutations that resulted in pigments, fragrances, or sugar-rich secretions became adaptive. For pollen that is carried by wind, the probability that any particular grain will actually land on a stigma is very low, whereas if it is carried by an insect that flies from flower to flower, the probability of a pollen grain reaching a stigma is much improved. Figure 9.25 ears of corn are really large groups of carplet flowers surrounded by protective leaf-like bracts. The corn silks are long styles. Corn tassels are inflorescences of stamenate flowers. Each corn plant produces both ears and tassels. The species is monoecious. When this insect-flower association began around 120 million years ago, neither insects nor flowers were particularly sophisticated. Insects probably visited all kinds of flowers, not recognizing the different species. As a result, pollen often landed on the stigma of the wrong species, where it was useless. Mutations in plants that increased a plant's distinctiveness, its recognizability by an insect, flower color, size, shape, fragrances, and so on, became selectively advantageous. Rewards for the insect such as sugary nectar or protein-rich pollen and stamens were advantageous. Mutations in insects were adaptive if they increased the capacity of insects to recognize flowers that offered abundant nectar or pollen. Because it is expensive energetically for an insect to fly, the ability to recognize the most nutritious flowers from a distance while flying is advantageous. As a result, many lines of insects and flowers underwent coevolution, a flower becoming adapted for visitation by a particular insect and the insect for efficient exploitation of the flower. Coevolution has also occurred between flowers and birds and between flowers and bats. The shape of the flower is particularly important, as a pollinator actually makes contact with it. Most flowers are radially symmetrical, that is, any longitudinal cut through the middle produces two halves that are mirror images of each other. These flowers are actinomorphic or regular. But all insects, birds, and bats are bilaterally symmetrical. Only one longitudinal plane produces two halves that are mirror images. In many species, flowers and pollinators have co-evolved in such a way that the flowers are now also bilaterally symmetrical, zygomorphic. When a pollinator approaches a zygomorphic flower, only one orientation is comfortable for it. Any misalignment prevents the pollinator's head or body from fitting the flower's distinctive shape. As a result, as the pollinator feeds at the flower, pollen is placed on a predictable part of its body. When it visits the next flower, pollen is rubbed directly onto the stigma. Not only is pollen carried to the appropriate flower, but it is carried directly to the stigmas. In contrast, a pollinator can approach an actinomorphic flower from any direction, and pollen may be brushed onto any part of the body. When it visits the next flower, the pollen-carrying part may miss the stigmas and cross-pollination will not occur. Figure 9.26. Any median longitudinal section of this poppy results in two halves that are mirror images of each other. It is radially symmetrical, that is, actinomorphic. These penstamen flowers are bilaterally symmetrical, zygomorphic, only if cut from top to bottom are the two halves mirror images. A pollinator, also bilaterally symmetrical, fits well only if it approaches the flower properly. Anthers are at the top of each flower's throat and will put pollen only onto a bee's back. Most plant-animal relationships are a battle. Animals try to eat plants, lay eggs in them, or do other harmful things. Plants defend themselves with poisons, spines, and scleroid barriers. Mutations that permit plants to make more effective deterrents benefit the plant but not its animal pests. But both plants and animals tend to benefit from the pollination relationship, and coevolution is possible. Mutations that make a plant more recognizable or more convenient to its pollinators help both the plant and the animal, just as certain mutations in pollinators are beneficial to both organisms. But neither side should give more than it has to. Some bees now are nectar robbers, being able to get nectar from flowers without having to carry any pollen. And a number of orchids, for example Chiloglottis, trick wasps into carrying pollen for free. These orchid flowers look, smell, and feel just like a female wasp, and male wasps try to mate with them. The wasps then fly off to the next orchid flower and the next, transferring pollen between each of them. The orchids get pollination without having to provide nectar, and the wasp gets nothing more than a good time. Wind-pollinated flowers. In species that are wind-pollinated, a totally distinct set of modifications is adaptive. Attracting pollinators is unnecessary, thus, mutations that prevent the formation of petals are selectively advantageous, and the energy saved can be used elsewhere in the plant. Sepals are also often reduced or absent, and the ovaries need no special protection, thus, the entire flower may be tiny. Zygomorphy provides no selective advantage. After pollen is released to the wind, the chance of any particular pollen grain landing on a compatible stigma is small, so huge numbers of grains must be produced. Large, feathery stigmas are adaptive by increasing the area that can catch pollen grains. In general, wind-pollinated individuals produce up to several thousand small flowers. Although each flower is tiny, the entire plant has a large total stigmatic surface area. Pollination is aided by the growth pattern of the plant population. Wind-pollinated species, like grasses, oaks, hickories, and all conifers, grow as dense populations in rangelands or forests. Within 1 kilometer may be found thousands of plants and, more importantly, millions of stigmas. Species that occur as widely scattered, rare individuals must rely on animal pollination. Ovary position. The ovary and ovules must be well protected from pollinators. Paradoxically, a flower must bring a hungry animal to within millimeters of protein-rich ovules in order to effect pollination. Adaptations that maximize the separation are long styles and stamen filaments. Burying the ovaries deep within the flower provides further protection. In some species, after all flower organ primordia are initiated at the receptacle apex, the primordia crowd together, and the bases of the stamens, petals, and sepals fuse, creating a thick layer of protective tissues around the ovaries, which appear to be located below the other organs. Two terms describe this. We can either say that the ovary is an inferior ovary or that the other parts are epigynous. Inferior ovaries also can result if receptacle tissue grows upward around the ovary. The more common arrangement, in which no fusion to the ovary occurs and the ovary is obviously above the other flower parts, is a superior ovary or hypogynous parts. Intermediate, partially buried ovaries are half inferior with perigynous flower parts. Figure 927. Many wind-pollinated trees have such tiny flowers people do not realize they are flowering plants at all. This is an entire set of flowers of poplar, each flower consisting of just numerous stamens. Inflorescences and pollination. The positioning of flowers on an individual plant is important. Few species have plants that produce only a single flower. Instead, many flowers are produced either within a single year or over a period of many years. A large mountain ash or cherry tree produces thousands of flowers every year for well over 100 years. Many important factors determine whether a flower is seen and visited by pollinators, for example, the positions of flowers relative to other flowers, leaves, and trunk, height from the ground, and distance from an open, uncluttered flight path for pollinators. Figure 928. In flowers with superior ovaries, carpels sit above the other organs, as would be expected because they are initiated last by the flower apical meristem. This is an inferior ovary resulting from the fusion of the bases of sepals, petals, and stamens to the base of the carpel. For appendages are involved whose vascular bundles are still present and distinct. In some species, after all of the appendage primordia are initiated, the receptacle tissue grows upward and surrounds the ovaries. This can be detected because the vascular bundles around the ovary are receptacle bundles, not appendage bundles. Reproductive success is measured in terms of the number of healthy, viable seedlings that become established. One important factor for this is pollination. It might seem optimal to have large flowers that can be easily seen by pollinators. Unfortunately, they can also be seen by herbivores, and because ovules and pollen are rich in protein, they are good food sources for pests. Production of the pedicel, receptacle, sepals, and petals can be thought of as packaging and advertising cost. The larger the flower is, the larger the cost. These costs can be made more acceptable by increasing the number of ovules per flower. Large flowers tend to have numerous ovules and small flowers fewer. Small flowers exist because small plants cannot afford large flowers and their accompanying large seeds, which are very expensive. Also, a large flower with many ovules is a big risk. If it is found and eaten, a major reproductive investment has been lost. However, damage to a small flower with only a few ovules is less significant. Also, if many flowers are grouped together, an inflorescence, they give a collective visual signal to pollinators. One small flower may be overlooked, but not 100 close together. Furthermore, in an inflorescence, the plant is able to control accurately the timing of the initiation, maturation, and opening of the flowers. Consequently, the plant can be in bloom and available to pollinators for several weeks even though each flower lasts only a day or two. Figure 929. Flowers of sausage tree are pollinated by bats, which do not like to fly among the clutter of leaves because their sonar does not work well there. Long stalks allow flowers to hang in open air where bats have free access to them. Angiosperms that grow underwater in streams and lakes usually have flowers that project above the water's surface, and are accessible to flying pollinators. Figure 930. Palm flowers are small and inconspicuous, but when grouped together, their inflorescence has even more impact than a single large flower. Thousands of tiny palm flowers are present here, each too small to be seen at this magnification. The inflorescence of combretum is even more striking than the individual flowers. Notice how the flowers mature and open sequentially. Large flowers can produce more nectar than small flowers, but this makes them a more tempting target for nectar robbers, animals that take nectar without carrying pollen. Furthermore, a flower should not produce enough nectar to completely satisfy even a legitimate pollinator because no incentive would remain for the pollinators to go to another flower. It is better if the pollinators get only enough to make them interested in searching for more. Many other factors affect the relative selective advantage or disadvantage of flower number and size, as well as the number of ovules per flower. Considering the numerous types of plants, pollinators, and environments, the diversity of flower and inflorescence types is not surprising. In the simplest arrangement, flowers occur individually in leaf axils or as a transformation of the shoot apex. When grouped into inflorescences, two basic arrangements occur: determinate inflorescences and indeterminate inflorescences. A determinate inflorescence has only a limited potential for growth because the inflorescence apex is converted to a flower, ending its possibilities for continued growth. Typically, but not always, the terminal flower opens first, and then lower ones open successively. In the simplest type, below the terminal flower is a bract with an axillary flower, which also may have a bract and axillary flower, and so on. In an indeterminate inflorescence, the lowest or outermost flowers open first, and even while these flowers are open, new flowers are still being initiated at the apex. A raceme has a major inflorescence axis, and the flowers are borne on pedicels that are all approximately the same length. Catkins are similar to racemes, differing in that the flowers are imperfect, either staminate or carplet, and all flowers of a single inflorescence are the same, and thus, each species must have both staminate catkins and carplet ones. Catkins almost always contain very small, wind-pollinated flowers. A spike is similar to a raceme except that the flowers are sessile, lacking a pedicel. A spadix is a spike-like inflorescence with imperfect flowers, but both types occur in the same inflorescence, most often with staminate flowers located in the upper portion of the inflorescence and carplet flowers in the lower portion, although they can intermingle. The main inflorescence axis is thick and fleshy with minute flowers embedded in it. The entire inflorescence is subtended or enclosed by a petal-like bract called a spatha. A panicle is a branched raceme with several flowers per branch. Figure 931 Inflorescence types: simple determinate inflorescence, compound determinate inflorescence, raceme, spike, umbel, head. Larger circles represent flowers that open earlier than those depicted as smaller circles. Several types of indeterminate inflorescences have no dominant main axis. In umbels, the inflorescence stalk ends in a small rounded portion from which arise numerous flowers. Their pedicels are long and arranged so that all flowers sit at the same height, forming a flat disc. A head is similar to an umbel except that the flowers are sessile and attached to a broad expansion of the inflorescence stalk. Numerous bracts may surround the inflorescence during development. Heads are almost synonymous with the aster family, sunflowers and dandelions being easily recognizable examples. In this group, the inflorescences are so compact and highly organized that they mimic single flowers. What appear to be the petals are really entire flowers, ray flowers, in which the petals are very large and fused together. The center of the inflorescence is composed of a different type of flower, disc flowers, in which the corollas are short and inconspicuous. Fruit types and seed dispersal. Fruits are adaptations that result in the protection and distribution of seeds. Many different agents disperse fruits and the seeds they contain. Gravity, wind, water, and animals are the most common. The principles involved in fruit function are somewhat opposed to each other. Fruits that are tough and full of fibers or scleriates, such as pecans, walnuts, Brazil nuts, and coconuts, offer maximum protection but are heavy and expensive metabolically. Also, the protected seed must be able to break out to make germination possible. A more fragile fruit is better for that. If animals are to disperse the seeds, part of the fruit must be edible or otherwise attractive, whereas the seed and embryo must be protected from consumption. A division of labor often occurs, some parts being protective, others attractive, and still others allowing germination. Table 9 4 Agents of dispersal. True fruits and accessory fruits. The term pericarp refers to the tissues of the fruit regardless of their origin. In most cases, this is the ovary wall, but in many species, especially those with inferior ovaries, the receptacle tissues or sepal, petal, and stamen tissues may also become involved in the fruit. The terms pericarp and fruit have been applied to both types of fruit. Thus, Now the term true fruit is used to refer to fruits containing only ovarian tissue. An accessory fruit is used if any non-ovarian tissue is present. Apples develop from inferior ovaries, and the bulk of the fruit is enlarged bases of sepals and petals. Only the innermost part is true fruit derived from carpels. Figure 932. The red, edible flesh of strawberry is really the receptacle, not carpel tissue. It is therefore an accessory fruit. The seeds are true fruits, each derived from one carpel of the flower. Pineapples develop from the coalescence of all the many true fruits of one inflorescence, so they are a multiple fruit. In addition, many non-carpellary tissues become involved, and are therefore accessory fruits as well. Bracts are visible here. Fusion of carpels also affects the nature of fruits. If the fruit develops from a single ovary or the fused ovaries of one flower, it is a simple fruit, the most common kind. If the separate carpels of one gynoecium fuse during development, an aggregate fruit results, such as raspberries. If during development all of the individual fruits of an inflorescence fuse into one fruit, it is a multiple fruit, as in figs, mulberries, and pineapple. These are also largely accessory fruits because in addition to the ovary tissue, the inflorescence axis, bracts, and various flower parts contribute to the mature fruit. Classification of fruit types. There are several ways of grouping or classifying fruits. In one method, emphasis is placed on whether the fruit is dry or fleshy. A dry fruit is one that is not typically eaten by the natural seed-distributing animals. Fleshy fruits are those that are eaten during the natural seed distribution process. A further classification of dry fruits emphasizes fruit opening. Dehiscent fruits break open and release the seeds, whereas indehiscent fruits do not. For the most part, fleshy fruits are indehiscent. Animals chew or digest the fruit, opening it. If uneaten, the fruit rots and liberates the seeds. Although many dry fruits are dehiscent, others must rot, or the seed must break the fruit open itself. Perhaps the simplest fruits are those of grasses. Each fruit develops from one carpel containing a single ovule. During maturation, the seed fills the fruit and fuses with the fruit wall, which remains thin. Seeds of corn and wheat are actually both the fruit and its single seed, fused into a single structure. It has little protection and no attraction for animals. The fruit seed falls and germinates close to the parent sporophyte. The fruits are indehiscent, and during germination, the embryo absorbs water, swells, and bursts the weak fruit. These fruits are caryopses. Table 9.5 Fruit types. Beans and peas form from a single carpel that contains several seeds. The fruits, known as legumes, are dry and inedible like caryopses but do not fuse to the seeds. At maturity, the two halves of the fruit twist and break open along the two specialized lines of weakness. After liberation from the legume, the seeds are protected from small animals and fungi by only the seed coat. Fruits or seeds carried by wind must be light. They often have wings or parachutes that keep them aloft as long as possible. Such fruits are dry and weigh much less than 1 gram. Fruits and seeds that are transported by water can be larger and heavier, but they must be buoyant and resist mildew and rot. An excellent example is coconuts that float from one island to another. Animals carry fruit in a variety of ways. Dry fruits with hooks or stickers catch onto animal fur or feathers. Sticky, tacky fruits glue themselves onto animals, and fleshy, sweet, colorful fruits are eaten. Edible fruits require particular specialization because the fruit must be consumed but the seeds must not be damaged. They must not be crushed by teeth or gizzard or be digested. Immature fruits deter frugivores by being hard, bitter, or sour, such as unripe apples or bananas. At maturity, the fruit is soft, sweet, flavorful, and typically strikingly different in color. The enclosed seeds often have hard seed coats or endocarps that allow them to pass through an animal unharmed. Dinosaurs may have been especially poor seed dispersers. They had no hair for hooks to tangle in, and their digestive tracts were often too long, hot, and fermentative for seeds to survive. Grapes and tomatoes are examples of simple fleshy fruits. They are berries because all parts of the fruit are soft and edible. The seed coat provides some protection against being crushed during chewing, but perhaps more important are the size of the seeds and their slippery seed coat, which allows the seeds to slip out from between our molars. Pomes differ from berries in two ways. First, they develop from inferior ovaries, and thus, the inner tissue is the true fruit and the outer tissue is accessory fruit. Second, the seeds are protected because the innermost fruit tissues, the cores, are bitter and tough. The most elaborate fleshy fruits are drupes, in which the innermost tissues, the endocarp, are extremely hard and totally inedible, full of sclerenchyma. The mesocarp becomes thick, fleshy, juicy, and sweet, and the exocarp forms the peel, whose color informs the frugivore of the fruit's ripeness. The endocarp of a drupe is known as the pit and is often mistaken for the seed, but the true seed and its seed coat are located inside. Drupes provide maximum attraction to animals with minimum danger to the seed. Because frugivores are a major danger to seeds, it is necessary to think about how edible fruits evolved. They must offer some advantage that outweighs the danger. Animals have predictable habits and migration patterns. Ants, birds, and mammals carefully select the sites where they feed, nest, and sleep. Therefore, seeds are not distributed at random but are moved through the environment to specific sites. One example is mistletoe. Its seeds are sticky and adhere to a bird's beak as it eats the fruit. The bird probably feeds and cleans itself in the same species of tree, thus, the seeds are rubbed off the beak directly onto a proper host tree. Few seeds are ever lost by dropping to the ground or being deposited in the wrong tree, as would happen with wind or water dispersal. Just as efficient is dispersal of fruits and seeds of marsh plants. The birds and animals that live in marshes typically spend little or no time in other habitats, so seeds carried by these animals are almost certainly distributed to favorable sites. Figure 933 at maturity, different layers in this dry fruit contract in opposite directions, causing the pod to twist and break open. As these maple fruits drop, the wings cause them to rotate and float, thus remaining aloft and traveling much farther on the wind. The spiral, hairy tails of these geranium fruits let them float on the wind over long distances. A special benefit of seed distribution by animals is that the deposited seed may find itself in a small mound of organic fertilizer. Seeds adapted to pass through an animal's digestive tract are resistant to digestive enzymes and can tolerate the high level of ammonium in fecal matter. Because many seeds of plants cannot tolerate these two factors, the adapted seeds find themselves in a microenvironment that is not only nutrient-rich but also excludes some competitors. Figure 934. This seedling is as happy as a clam at high tide. It has enough fertilizer to last for months. Many of its competitors have been digested, others are being suffocated. At the next level. 1. Pollination syndromes. Each type of pollination agent affects the evolution of the flowers it pollinates. Wind-pollinated flowers usually evolve to lack sepals and petals. Hummingbirds prefer flowers that are long, tubular, and red. Bats fly at night and bat-pollinated flowers evolve to open after sunset, to have a strong fragrance, and even to reflect the bat's sonar back to the bat such that the flower is found easily in the dark. Dozens of other pollination syndromes occur and are remarkable examples of coevolution between plants and animals. Be sure to search for pseudocopulation between wasps and orchids. 2. Frugivores and seed dispersal. Coevolution has occurred between plants and animals such that seeds are dispersed to preferred sites rather than at random. For example, seeds of epiphytic plants and parasitic plants should be carried to new branches of suitable host trees, usually by having birds eat the fruits without damaging the seeds. As with pollination, many syndromes have evolved, some involving fruit as a reward for dispersal, others involving food bodies on the seeds themselves. Of special interest, several plant species produce edible fruits, but we have not found any animals that eat them. One hypothesis is that the animals went extinct recently, but the plants persist even though their seeds are no longer being dispersed effectively. Be sure to search on extinct seed dispersers. 3. Evolution of flowers. In most flowers, both stamens and carpels occur together within the same flower. Perfect flowers are common and evidence indicates that the earliest flowers were perfect. But in all other seed plants, pollen cones and seed cones are always separate. Pollen-producing structures never occur together with ovule-producing structures. Thus, we are still far from being certain of the steps involved in the evolution of early flowers, the early angiosperms, and their ancestors, but this is a fascinating area. There are conflicting theories and conflicting evidence. This is a challenging topic for anyone, especially for beginning botany students, but it is very rewarding. 4. Recommended books. A team of botanists and a visual artist created 3 wonderful books about plant reproduction. These are large coffee table books with large illustrations, each image being a photograph or a scanning electron micrograph of pollen or fruits or seeds. SEM images are naturally black and white, but the team colorized the images to enhance certain features so that the various parts are more readily understandable. Each is accompanied by just enough text to fully explain the biology without overwhelming the reader. I recommend them highly. Pollen, The Hidden Sexuality of Flowers by Rob Kessler and Madeline Harley. Fruit, Edible, Inedible, Incredible by Wolfgang Stuppy and Rob Kessler. Seeds, Time Capsules of Life by Rob Kessler and Wolfgang Stuppy. Summary. 1. Asexual reproduction results in new individuals genetically identical to the parent. The parent and all progeny are equally adapted to the habitat. 2. Sexual reproduction results in progeny that differ from each other genetically. There is a range of fitness, and some progeny may have a combination of maternal and paternal genes that causes them to be even better adapted than the parents. 3. All seed plants have a life cycle that consists of an alternation of heteromorphic generations. Microgametes and megagametes are produced by microgametophytes and megagametophytes that grow from microspores and megaspores. Most species have only one type of sporophyte, capable of producing both types of spores. Dioecious species have 2 types of sporophytes, one of which produces microspores and the other megaspores. 4. Flowers contain all or some of the following: non-essential organs, pedicel, receptacle, sepals, and petals, and essential organs, stamens and carpels. 5. In flowering plants, the microgametophyte consists initially of a vegetative cell and a generative cell, but the latter divides into 2 sperm cells. The megagametophyte consists of the egg cell, 2 synergid cells, a binucleate central cell, and 3 antipodal cells. 6. After fertilization, the zygote grows into an embryo. Polar nuclei fuse with the second sperm nucleus, establishing the endosperm nucleus and endosperm, and the integuments develop into a seed coat. The entire structure is a seed. The ovary develops into a fruit. 7. Inflorescences are groups of many flowers. They may be determinate or indeterminate and may contain several types of specialized flowers. 8. Fruits protect seeds during development and may aid in their dispersal and maturation. Fruits are either dry, inedible, dehiscent, or indehiscent, or they are fleshy, edible, and indehiscent. Important terms. Alternation of generations, anther carpels, coevolution, compatibility barriers. Complete flowers, cotyledons, cross-pollination, double fertilization, embryo, sac, endosperm, epicotyl, fruit, gametes, gametophyte. Heteromorphic generations, hypocotyl, imperfect flowers, incomplete flowers, inflorescence, karyogamy, ovary, ovules, pedicel, perfect flower, petals, plasmogamy, pollen, pollen tube, radicle, receptacle, seed coat, seed, sepals, spores, sporophyte, stamens, stigma, style, syngamy, zygote. Review questions. 1. Reproduction can serve 2 very different functions. What are they? 2. Under what conditions is it selectively advantageous for a plant to produce offspring identical to itself? 3. Imagine a plant that has been well adapted to a particular habitat, and it has been reproducing only asexually, thus, all of its offspring are identical to it. If the climate or other conditions change such that the plant can no longer survive in the new conditions, what will happen to all of its offspring? Why? 4. With asexual reproduction, are progeny ever more fit, more adapted than the parent? 5. Look at the apples in Figure 9.1a, all growing on a single tree. Do all of the seeds in all of the apples in the photograph have the same maternal parent? Do they all have the same paternal parent? 6. Sexual reproduction produces offspring that are not identical to each other or to either parent. Usually, some are more well adapted than the parents. Some are more poorly adapted, and most are about as adapted. What is one of the beneficial aspects of this diversity? 7. In stable populations, ones that are neither increasing nor decreasing in abundance. About how many of a plant's seed survive and grow to adulthood, being able to replace it when it dies? If during the plant's lifetime it produces 100,000 seeds, how many do not survive, do not grow, and cannot replace it when it dies? 8. Describe the life cycle of us humans. Are the tissues and organs in our bodies made up of diploid cells or haploid ones? Our reproductive organs make sperm cells in males and egg cells in females. Is this done by mitosis or meiosis? When our bodies make gametes, does every cell in our body become a sperm cell or an egg cell, or do just some of the cells in our reproductive organs do this? Can our haploid sperm and egg cells undergo mitosis and grow into new animals that look like us but are haploid instead? After a fertilized egg is formed, is it diploid or haploid? Can it immediately undergo meiosis to make 4 new sperm cells or egg cells, or can it only grow by mitosis into another person? 9. The plants we are familiar with are called sporophytes or the sporophyte generation. Do these plants have bodies made up of diploid cells or haploid ones? In which organs does meiosis occur, leaves, stems, stamens, roots, carpels, or petals? When plants undergo meiosis, do all cells of the plant undergo meiosis? Do all cells become haploid, or does meiosis occur in just a few cells in some flower parts? 10. In animals, meiosis produces gametes, but that does not happen in plants. When some of the cells of a sporophyte undergo meiosis, what types of cells are produced? What do they grow into? 11. What is a gametophyte? How many different types of gametophytes are there in a plant life cycle? What do they look like? Do the sporophytes and gametophytes of seed plants ever look like each other? 12. Draw and label a microgametophyte of a flowering plant. What type of gamete does it produce? Draw and label a megagametophyte of a flowering plant. What type of gamete does it produce? 13. Flowers typically have many parts, although some flowers can be missing some of the standard parts. What is the name of each of the following parts? The stalk of the flower. B. The end of the stalk, where the other parts are attached. C. The parts that are usually green and protect the flower bud as it develops. D. The parts that attract pollinators. E. The parts that produce pollen. F. The parts that receive pollen and which contain ovules. 14. If a flower has all of the parts C, D, E, and F in question 13, they are said to be flowers. If they are missing any of these parts, they are said to be flowers. Parts E and F are especially important because they produce the reproductive cells, the spores. If a flower has both E and F in question 13, they are flowers. If they are missing either E or F or both, they are flowers. 15. In flowers that are pollinated by wind or water, which of the parts in question 13 is often missing? 16. What is the difference between a perfect and an imperfect flower? What is the difference between a complete and an incomplete flower? 17. A stamen usually has 2 parts. The stalk is called A and an upper portion, the, which produces the pollen. Only some of the cells in the upper part undergo meiosis and become pollen grains. Those cells are called mother cells or neighboring anther cells, in a layer called the act as nurse cells. 18. Carpels usually have 3 parts, a that catches pollen grains, a that elevates the first part, and an where megaspores are produced. In this last part, there are placentae that bear small structures called each with a short stalk called a funiculus and a central mass of parenchyma called the nacellus. One cell in the nacellus will be the mother cell or 19. The megaspore in most flowering plants grows into a megagametophyte that has 7 cells and 8 nuclei. Name and describe the 7 cells. 20. After pollen lands on a stigma, it is far away from the ovule with the megagametophyte, which holds the egg. How are the 2 sperm cells transported from the stigma to the egg? 21. In angiosperms, as a sperm cell enters the egg, it loses both and such that only the sperm cell nucleus contributes any DNA to the new zygote. In gymnosperms, however, the sperm cell loses only its such that the zygote inherits both plastids and a nucleus from the sperm cell. 22. After the sperm cells enter a synergid, one fertilizes the egg in a 2-step process. First there is fusion of the sperm cell's protoplasm with that of the egg, a step called. Then the sperm cell nucleus fuses with the egg cell nucleus, the second step, called. 23. What happens to the second sperm nucleus, the one that does not fertilize the egg cell? What is the tissue that develops from this second fertilization? How is coconut related to this? 24. In most eudicot seeds, the parts of the embryo are very easy to see. Describe each of these parts. Cotyledons: B. Radical. C. Hypocotyl. D. Epicotyl. E. Extra credit portion. Answer the following question if you have read about the radicle with regard to a taproot system and a fibrous root system. How is the radicle involved in these different types of root systems? 25. What are albuminous and exalbuminous seeds? Consider corn, peas, and beans. Which of these seeds are which? 26. After pollination and fertilization, as the ovule develops into a seed, the ovary matures into a. Many of these have 3 parts. The is the skin or peel. The is the flesh, and the innermost layer, the may be tough like the pit of a cherry. 27. What is the difference between self-pollination and cross-pollination? If pollen is transferred from the stamens of a flower to the stigma of the very same flower, is that cross-pollination or self-pollination? If it is transferred from the stamen of one flower to the stigma of another flower on the very same plant? If it is transferred from the stamen of one flower to the stigma of another flower on a different plant, but a plant that is a clone of the first one? Animals do not have pollen, of course. But why is it that most animals never have to worry about the equivalent problem of self-fertilization? Why cannot most animals fertilize themselves? 28. Describe a species that is dioecious. Name several examples. In order for sexual reproduction to occur in a dioecious species, how many separate plants must be involved? Name them all. 29. Describe a monoecious species. Name several examples. 30. In several pairs of plant and animal species, plants have become modified such that only its animal partner can pollinate it, and the animals have become modified such that they are especially well adapted to pollinate just their plant partner. What is the name of this type of evolution that results in 2 organisms becoming particularly adapted to and dependent on each other? 31. Explain the following terms: inferior ovary, superior ovary, actinomorphic flower, zygomorphic flower. How is each of these modifications selectively advantageous? 32. What is an inflorescence? The inflorescences of a sausage tree hang far down out of the tree. How is this of benefit to bats? Why can the inflorescence of combretum attract more pollinators than can the individual flowers? 33. After pollination and then fertilization, what usually happens to each of the following: stigma, style, carpel, ovule, integuments, and zygote? 34. Fruits are often classified as dry or fleshy. What is the difference? Which of these 2 are dehiscent, which are indehiscent? 35. In ordinary English, we use the word fruit to mean something sweet and juicy. However, the following things are fruits: peanut shells, pea pods, bell peppers, and chili peppers. What is the characteristic that lets us know these really are fruits even though they are not sweet? In contrast, bananas are fruits that do not have this characteristic. 36. Some things that we call fruits are not true fruits but instead are accessory fruits. What is the red part of a strawberry, and what are the true strawberry fruits? In an apple, what is the fleshy part that we eat, and what is the core that we throw away? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Part 2. Plant Physiology and Development. Chapter 10 Energy Metabolism, Photosynthesis. Chapter 11 Energy Metabolism, Respiration. Chapter 12 Transport Processes. Chapter 13 Soils and Mineral Nutrition. Chapter 14 Development and Morphogenesis. Chapter 15 Genes and the Genetic Basis of Metabolism and Development. Part Opener Image. Plants often develop in wonderfully diverse ways. The main branch here grows horizontally, but all its branches grow upward. Notice too that only axillary buds on the upper surface of the horizontal branch have grown out, whereas all those on the sides and lower surface are still dormant. All axillary buds in the vertical branches are dormant. As these branches grow, they perceive and respond to conditions around themselves. Almost certainly the important environmental cue is either sunlight or gravity. Can you design an experiment to determine which factor is controlling the physiology of these branches? Araucaria subulata. When you are studying plant structure, you can often see the material directly with the naked eye, although light or electron microscopy may be necessary to see some structures. In plant physiology and development, however, the objects of our study, chemical reactions and metabolic pathways, cannot be seen at all. Instead, the results of experiments, measurements, and analyses are studied, and hypothetical reactions and pathways are set down on paper. From these, predictions are made and new observations planned to test the hypotheses. It is easy for us to study and memorize metabolic pathways, chemical formulas, and diagrams of physiological control mechanisms without appreciating that these have never been, and cannot ever be, seen directly. Every one is a theoretical model that is consistent with the majority of the available data and that is logically and internally consistent. When experienced anatomists see an unusual structure, they may be able to recognize instantly that it is new to science or is at least a significant variation. For physiologists, it is not so easy. If an experiment on photosynthesis does not come out as expected, it may be that a new type of photosynthesis is being discovered. It may be that the current theories of photosynthesis are not completely accurate and have made an erroneous prediction, or it may be that an experimental error has occurred. It can be difficult for students to appreciate the tremendous amount of careful, ingenious work that must be done just to establish that a particular theoretical metabolic pathway truly represents the reactions that occur in certain plants. Whereas it is relatively easy to determine that natural selection has resulted in the evolution of many types of leaves, stems, roots, flowers, xylem, phloem, and so on, it is much more difficult in physiology. Numerous differences in microhabitats, water availability, heat, cold, soils, pests, and plant diseases have resulted in diverse types of structures that are selectively advantageous under various conditions. It is logical to expect the same to be true of metabolism. We do know that there are several varieties of photosynthesis and respiration, and there may be others that have not yet been discovered. As you study the material in this section, keep in mind that, just as is true for structure, organization is of fundamental importance for metabolism. The chemical and physical reactions that constitute plant metabolism are highly ordered and not at all random. This orderliness is maintained by the input of energy acting on materials brought into plants from the environment. There are many types of order, and the information necessary to establish the proper reactions acting on the proper material is stored in the genes, both in the nucleus and in the plastids and mitochondria. The mechanisms by which the genes control the interaction of energy and matter, such that a plant of a particular species results, are discussed in chapters 14 and 15. Chapter 10. Energy Metabolism, Photosynthesis. Chapter Opener Image. The high-energy electrons moving through these power lines are dangerous and could easily kill a person. Photosynthesis also involves the movement of high-energy electrons, which, if not controlled properly, can and do damage chloroplasts and even the whole plant. There was no electricity flowing through these lines when they were being constructed, but a plant's photosynthetic pigments become energized as soon as they are synthesized, and they are even dangerous in autumn while chloroplasts are being dismantled. Whenever light strikes chlorophyll, an essential but risky process begins. Outline. Concepts. Energy and reducing power. Energy carriers. Reducing power. Other electron carriers. Photosynthesis. The light-dependent reactions. The stroma reactions. Anabolic metabolism. Environmental and internal factors. Light. Leaf structure. Water. C metabolism. Crassylation, acid metabolism. Box 10-1 Plants and people, photosynthesis, global warming, and global climate change. Box 10-2 Alternatives, photosynthesis in bacteria and cyanobacteria. Learning objectives. After reading this chapter, students will be able to recall the method of energy transportation from energized pigments into endergonic reactions. Explain why plants need reducing power. List 4 important qualities of carbon dioxide and water as photosynthesis compounds. Describe the ideal photosynthetic pigment. Compare chlorophyll A to this ideal. Summarize the function of accessory pigments. Describe the ways in which the light-dependent reactions and the stroma reactions function together. Explain the ways in which photosynthesis affects Earth's climate. Did you know? The photosynthetic pigment chlorophyll absorbs mostly red and blue but is poor at absorbing green light. Green light bounces off and thus leaves appear to be green. Almost all energy used by animals, fungi, and everything else is the energy of sunlight captured by photosynthesis in plants, algae, and cyanobacteria. Light intensity must be just right. In habitats where sunlight is too intense, plants often have hairs or spines that shade the plant, if light is too dim. Not enough energy is available to allow life. Concepts. Probably the most important concept concerning cells and all of life itself is that living organisms are highly ordered, highly structured systems. The universe as a whole is constantly becoming less orderly, its disorder is increasing. Prokaryotes, protists, fungi, plants, and animals, however, represent phenomena in which particles become more orderly. A plant absorbs diffusely scattered molecules of carbon dioxide, water, and minerals and organizes them into organic molecules, cells, tissues, and organs. Each plant carries this out with such precision that each species of plant is easily distinguishable from others. After death, decay is the process by which an organism's molecules become more disordered and scattered, their entropy increases. Because living organisms are part of the natural world described by the laws of physics and chemistry, the decrease in the entropy of living organisms must obey physical laws. This is accomplished by putting energy into the living system, the source of energy being sunlight. To be accurate, we must consider the sun and life together. Atomic reactions that generate sunlight cause greater disorder in the sun than sunlight causes order in living organisms. The entire system becomes more disordered. Because there is no means of putting energy into an organism's body after death, an increase in entropy cannot be prevented. Sunlight maintains and increases the orderliness of life by two methods: directly, in the process of photosynthesis, which produces complex organic compounds, and indirectly, in the respiration of those organic compounds, either by the organism itself or by another organism that eats it. These two methods of supplying energy and maintaining orderliness, photosynthesis and respiration, are the basis for a major, fundamental distinction in the types of organisms. Photoautotrophs are organisms that gather energy directly from light and use it to assimilate small inorganic molecules into their own tissues. Photoautotrophs include all green plants, all cyanobacteria, and the few bacteria capable of photosynthesis. Heterotrophs are organisms that cannot do this but instead take in organic molecules and respire them, obtaining the energy available in them. Heterotrophs include all animals, all completely parasitic plants, all fungi, and non-photosynthetic prokaryotes. Gathering energy by taking in organic material has the advantage that part of the material can be used as construction material instead of fuel. At least some of the amino acids, fatty acids, and sugars in food can be built into the organism's own polymers and the rest respired for energy. Photoautotrophs must build all of their own molecules using just carbon dioxide, water, and various nitrates, sulfates, and other minerals. Tremendously important consequences follow from the fact that photoautotrophs and heterotrophs differ in their sources of energy and building material. Sunlight and carbon dioxide do not need to be stalked, hunted, and captured, thus, sensory organs, muscles, and central nervous systems like those of animals are unnecessary. Conversely, the ocean is full of microscopic bits of food, and animals such as sponges and corals can gather it the way plants gather carbon dioxide. The mode of nutrition has had overriding influence on the bodies and metabolisms of plants and animals. Tissues and organs are also either photoautotrophic or heterotrophic. Chlorophyllous leaves and stems are photoautotrophic, whereas roots, wood, and flowers are heterotrophic and survive on carbohydrates imported through phloem. During winter, if all leaves have upsized, the entire plant may be composed of heterotrophic tissues, and it maintains its metabolism by respiring stored starch. Figure 10.1 All organisms must obtain the energy needed to drive their metabolism. Most plants obtain energy through photosynthesis using the green pigment chlorophyll. All animals and fungi obtain their energy by either eating plants or by eating animals that ate plants. Some plants, called holoparasites, like this orange dodder, do not carry out photosynthesis. Instead, they send specialized roots called haustoria into the bodies of ordinary green, photosynthetic plants and extract sugars, water, and minerals from them and convert the sugars into the organic compounds they need. Close-up showing haustoria penetrating the leaf of a host. Table 10.1 Differences between photoautotrophs and heterotrophs. Tissues often change their type of metabolism. Young seedlings are white and heterotrophic while germinating underground. They survive on nutrients stored in cotyledons or endosperm. Seedlings become photoautotrophic only after they emerge into sunlight. Immature fruits may be green and photosynthetic, however, in the last stages of maturation, chloroplasts are converted to chromoplasts, and metabolism then depends on imported or stored nutrients. Chromoplasts are plastids that contain large amounts of pigments other than chlorophyll. Young leaf primordia are green, but they grow more rapidly than their own photosynthesis would permit. They have a mixed metabolism of photosynthesis and carbohydrate import. Photosynthesis is a complex process by which carbon dioxide is converted to carbohydrate. This involves endergonic reactions driven by adenosine triphosphate and requiring new bonding orbitals filled by electrons carried to the reaction by reduced nicotinamide adenine dinucleotide phosphate. Before this can happen, ATP and NADPH themselves must be formed in highly endergonic reactions driven by light energy. In order to understand this, you must first understand the nature of light and pigments along with the concept of reducing power. Energy and reducing power. Energy carriers. Energy enters the biological world through photosynthesis, a process that converts light energy to chemical energy. The sun's light is captured by certain plant pigments that use the energy in chemical reactions. Unfortunately, the energized pigments can enter into only 2 chemical reactions, although plants have thousands of different reactions in their metabolism. Several theoretical ways exist of transporting energy from energized pigments into endergonic reactions. Figure 10.2 The tail of chlorophyll contains only hydrogen and methyl functional groups, so it is hydrophobic and dissolves into the chloroplast's membrane lipids, immobilizing it. The porphyrin ring system of alternating double and single bonds acts as an antenna that captures light energy. The magnesium atom carries the electrons involved in photosynthesis. If the colored methyl group were an aldehyde group, the pigment would be chlorophyll B. 1. Allow the pigments to enter into every reaction necessary. A problem is that the energized pigments are large molecules, therefore, they are not very mobile and never move across membranes. Furthermore, they are too energetic. They can react with almost anything and would be difficult to control. Table 10. 2 methods of synthesizing ATP. 2. Allow the energized pigments to make one or several smaller, less energetic intermediates that can be moved and controlled easily. Such a method has evolved. Photosynthetic reactions produce ATP, an extraordinary molecule. Its high-energy phosphate bonds carry enough energy to force almost any reaction to proceed, and it can enter into almost every reaction for which energy is needed. In those that it does not enter, other energy carriers, often relatives of ATP, are involved. The most frequent is guanosine triphosphate, which also carries high-energy phosphate bonds. Although ATP is an essential molecule, it constitutes only a tiny fraction of the plant body. Each molecule is recycled and reused repeatedly, thousands of times per second. ATP is converted to ADP and phosphate by metabolic reactions, but the phosphate can be reattached with a high-energy bond by the reactions of either photosynthesis or respiration. Each molecule is an energy carrier, shuttling between reactions that release energy and those that consume it. There are 3 methods by which adenosine diphosphate can be phosphorylated to ATP. The first, photophosphorylation, involves light energy in photosynthesis. Animals, fungi, and non-chlorophyllous plant tissues cannot perform photophosphorylation because they lack the necessary pigments and organelles. Instead, they respire some of the high-energy compounds they have consumed as food or imported by phloem. Compounds with high-energy phosphate groups are produced, and these compounds force their phosphate onto ADP, making ATP. This is substrate-level phosphorylation. In the last stages of respiration, ADP is phosphorylated to ATP by oxidative phosphorylation. Each process occurs in a distinct site within the cell, and each captures energy from distinct types of exergonic reactions. Photophosphorylation occurs only in chloroplasts in light, but substrate-level and oxidative phosphorylation occur in all parts of the plant at all times. Reducing power. Earth's atmosphere is about 21% oxygen, therefore, many compounds are found in their oxidized form, carbon as carbon dioxide, sulfur as sulfate, nitrogen as nitrate, and so on. Oxidized means that an atom does not carry as many electrons as it could. In carbon dioxide, each oxygen can be considered to have pulled 2 electrons almost completely away from the carbon, and the carbon is said to be at a +4 oxidation state. This is speaking figuratively. Electrons spend more time near the oxygen than they do near the carbon, sulfur, or nitrogen, but they are not torn completely away. These bonds are covalent, not ionic. When electrons are added to an atom, it becomes reduced. Think of a reduction reaction as one that reduces the positive charge on an atom and an oxidation reaction as one that increases the positive charge. A preliminary rule of thumb is that oxidized compounds often contain a great deal of oxygen, whereas reduced compounds contain hydrogen. Oxygen has a strong tendency to pull electrons away from an atom and raise that atom's partial positive charge, but hydrogen becomes more stable by giving up electrons, reducing its partner's partial positive charge. Electrons can be transferred only between atoms or molecules, so each reaction in Table 10.4 is only a half reaction. Every oxidation occurs simultaneously with a reduction. The full reaction is known as an oxidation-reduction reaction or redox reaction. Table 10.3 Calculation of oxidation states. Table 10.4 Reductions and oxidations. Whereas compounds in the environment are predominantly in the oxidized state because of our oxygen-rich atmosphere, most compounds in organisms are in the reduced state. Carbon is often in the form of carbohydrate, where its oxidation state is +0, nitrogen is frequently present as an amino group, NH, and sulfur is present as SH. Thus, in addition to needing energy, organisms also need reducing power, the ability to force electrons onto compounds. Reducing power is especially important to plants because they take in carbon dioxide and water. the most highly oxidized forms of carbon and hydrogen, and then convert them to carbohydrates, fats, and other compounds that are very reduced. Heterotrophs have less of a problem. When they consume plants, they get compounds that have already been reduced. They do need some reducing power, however, when they synthesize compounds that are very reduced, such as fatty acids. Just as with energy, an optimum solution for moving and handling reducing power, electrons, is to use small molecules that are semi-stable and mobile. The two molecules used most often are nicotinamide adenine dinucleotide and the closely related nicotinamide adenine dinucleotide phosphate. Both can pick up a pair of electrons and a proton, thereby becoming reduced to NADH and NADPH. When they reduce a compound by transferring their electrons to it, the proton is released and NAD or NADP is regenerated. Rather than having a large number of carrier molecules, the cell recycles each molecule, using it thousands of times a second as it shuttles between electron-producing and electron-consuming reactions. Because NAD and NADP take electrons away from other molecules, they are oxidizing agents. They oxidize the material they react with. It is also possible to say that the material has reduced the NAD or NADP. During the process, NADH and NADPH, two strong reducing agents, are formed. They have a powerful tendency to place electrons onto other molecules, reducing those molecules and becoming oxidized themselves. The tendency to accept or donate electrons varies greatly and is known as a molecule's redox potential. Cells contain a variety of electron carriers that differ in their tendency to accept or donate electrons. Figure 10.3 here, only the portion of NAD involved in carrying electrons is shown. The oxidized state carries a partial positive charge on the nitrogen and 3 double bonds in the ring. When it is reduced by 2 electrons, the positive charge disappears from the nitrogen, numerous bonding orbitals within the ring are changed, and the top carbon picks up a proton. No bonding orbitals are formed between NAD and the electron donor, therefore, NADH is free to diffuse away after picking up electrons. Similarly, as it donates electrons to some substrate, the ring bonding orbitals revert to the NAD form, none of which binds it to the substrate, so the NAD diffuses away. The same is true for NADP. In the upper reaction, 2 electrons are passed from a hypothetical molecule AH to an ADP. AH has been oxidized to A, and NADP has been reduced to NADPH plus H. The NADPH is free to diffuse to another site, where it passes the 2 electrons onto another hypothetical molecule, B, reducing it to BH and becoming oxidized back to an ADP. Table 10.5 Redox potentials of electron carriers. Compounds with a large negative redox potential tend to donate electrons and exist in the oxidized state, X, ferredoxin, NADP, FAD. Compounds with a large positive redox potential tend to accept electrons and exist in the reduced form, Cita plus 2, HO. Other electron carriers. Cytochromes. Cytochromes are small proteins that contain a cofactor, heme, which holds an iron atom. The iron carries electrons and cycles between the +2 and +3 oxidation states. Cytochromes are intrinsic membrane proteins. They are an integral part of the chloroplast's thylakoid membranes and cannot be removed without destroying the membrane. Consequently, they carry electrons only between sites that are extremely close together within a membrane rather than diffusing throughout the stroma as NADPH does. Plastoquinones. Plastoquinones, like cytochromes, transport electrons over short distances within a membrane. After they pick up 2 electrons, they also bind 2 protons. Their long hydrocarbon tail causes them to be hydrophobic, so they dissolve easily into the lipid component of chloroplast membranes. Plastocyanin. Like cytochromes, plastocyanin is a small protein that carries electrons on a metal atom, in this case copper. When oxidized, the copper ion is in the +2 oxidation state, but as it picks up the electron, it is reduced 1 level to the +1 oxidation state. Plastocyanin is loosely associated with chloroplast membranes. It can move a short distance along the surface, but it does not travel far. Figure 10.4 In cytochromes, Iron is not bonded directly to any amino acid but is held by heme, a box-like porphyrin ring similar to the portion of chlorophyll that holds the magnesium ion. The distinguishing feature of the quinone class of electron carriers is that each has two ketone groups whose carbon atoms are part of a ring structure. Photosynthesis. As the name implies, photosynthesis is a process that uses light energy to synthesize something. The term is so general that it could be applied to many different types of reactions, but whenever a botanist uses the term photosynthesis, the reaction being discussed is the combination of carbon dioxide with water to form carbohydrate, figure 10. Think about why these particular compounds are part of photosynthesis. First, both carbon dioxide and water are abundant and cheap, occurring almost everywhere in large quantities. The exception is the lack of water in severe deserts like the Sahara, where very little life exists simply because water is scarce. It is important to have a metabolism based on abundant compounds. It is necessary also for raw materials to be cheap, that is, the plant must be able to obtain them without expending much energy. Water and carbon dioxide are excellent because they diffuse into the plants automatically from soil, air, or water. Another important quality of carbon dioxide and water is that they are very stable and contain little chemical energy, so it is possible to deposit a large amount of energy into them. The carbohydrates they form are a good means of storing energy because all reactions leading to carbohydrate breakdown have high energy of activation barriers. Despite being energy-rich, carbohydrates are stable and chemically unreactive. Finally, both the reactants and the products of photosynthesis are non-toxic. It is safe to absorb large quantities of carbon dioxide and water and to store high concentrations of carbohydrates. Many substances that are critical for life are extraordinarily toxic if they become even slightly concentrated. Chlorine, sodium, ammonium, and many vitamins are just a few examples. Figure 10.5. Although this chemical equation succinctly summarizes photosynthesis, it reveals virtually nothing of the reaction mechanism or the many carriers and enzymes that participate. We cannot draw a reaction diagram because photosynthesis does not occur by the direct interaction of 6 molecules of carbon dioxide with 6 of water. However, the relative potential energies can be shown, indicating that this is an endergonic process. During photosynthesis, the carbon of carbon dioxide is reduced and energy is supplied to it, converting it to carbohydrate. The carbon atom in carbon dioxide is at the +4 oxidation state, whereas carbon atoms in carbohydrate are, in general, at +0. 4 electrons must be found and placed into new bonding orbitals around the carbon atom to reduce it. This is not easy because carbon is more stable in the oxidized state than in the reduced state. Carbohydrates such as wood and sugar can burn, releasing energy, but carbon dioxide does not. Therefore, a source of electrons and a source of energy are necessary for photosynthesis. The electron source is water, and the energy source is light. Water and light, however, do not act on carbon dioxide directly. Instead, they create the intermediates ATP and NADPH by a process called the light-dependent reactions. In a separate set of reactions, the stroma reactions, ATP and NADPH interact with carbon dioxide and actually produce carbohydrate. Figure 10.6. Light-dependent reactions of photosynthesis occur by means of membrane-bound carriers, but the actual formation of carbohydrate occurs in the chloroplast liquid. ATP-ADP and NADP-NADP diffuse between the two regions. No region of the chloroplast is far from a membrane, so the distances traveled are only a few hundred times the diameter of a molecule. Figure 10.7. On the left end of this electromagnetic spectrum are gamma rays. If treated as waves, they have extremely short wavelengths, less than 1 nanometer. If treated as particles, each quantum is highly energetic. Moving to the right, wavelengths become longer and quanta less energetic. The region to which our eyes respond, the visible region, is enlarged. These apples have an abundance of pigments that absorb all colors except red. Earlier, while still developing, they had the pigment chlorophyll, which absorbs red and blue strongly, but reflects green. The light-dependent reactions. The nature of light. Light is one small segment of the electromagnetic radiation spectrum, which encompasses gamma rays, X-rays, ultraviolet light, infrared light, microwaves, and radio waves, in addition to visible light. Radiation can be thought of and treated physically either as a set of particles called quanta, also called photons, or as a set of waves. The various types of radiation differ from each other only in their wavelengths and the amounts of energy each individual quantum contains. Short wavelengths have relatively large amounts of energy in each quantum, whereas long wavelengths have relatively little. Because humans use visible light for vision, we are more sensitive to and familiar with this region of the spectrum. We cannot see other wavelengths, but we can feel infrared radiation as heat, and our bodies react to ultraviolet light by becoming tanned or burned. We distinguish differences in quantum energy of visible light as differences in color. Most of us see all wavelengths from red through orange, yellow, green, blue, indigo, to violet. Certain insects see some near ultraviolet, but in general, all animals see in the range from 350 to 760 nanometers, which is also the radiation that plants use for photosynthesis. The nature of pigments. Most materials absorb certain wavelengths more than other wavelengths. If a substance absorbs all wavelengths except red, then red light either bounces off or passes through it, and the substance appears red to us. Any material that absorbs certain wavelengths specifically and therefore has distinctive color is a pigment, but we more often think of pigments as substances that absorb light as part of their biological function. Some pigments, such as melanin, absorb light and thereby protect other light-sensitive substances. Other pigments, such as those in flowers, fruits, or the skins of animals, are important for the light they do not absorb, which gives the pigments their color, allowing them to be useful to the organisms in attracting mates, pollinators, or frugivores or in hiding from predators. Photosynthetic pigments transfer absorbed light energy to electrons that then enter chemical reactions. Because only absorbed energy can be used, a theoretically ideal photosynthetic pigment would be black. It would absorb and use all light, not letting any escape. The pigment should at least absorb high-energy radiation instead of the fairly weak visible light. Rather surprisingly, the critical pigment, chlorophyll A, is not like this at all. It absorbs only some red and some blue light, letting most of the rest pass through, especially high-energy radiation. In general, plants only capture about 5% of the energy available in the electromagnetic radiation that strikes them. Why does chlorophyll A lack what seem like ideal characteristics? Plants and people. Box 10-1 Photosynthesis, global warming, and global climate change. Our atmosphere is critically important to life on Earth. Ironically, its composition is the product of that very life. The free oxygen we breathe is produced solely by oxygenic photosynthesis. There is no other source. Oxygenic photosynthesis originated 2.8 billion years ago. We know this because for millions of years the newly produced oxygen reacted with iron, forming a worldwide stratum of rust in ancient rocks. After all iron had been oxidized, free oxygen began accumulating in the atmosphere, and its concentration has been increasing ever since. Simultaneously, photosynthesis pulled carbon dioxide out of the atmosphere, converting it first to 3-phosphoglyceraldehyde and then to all of the other organic compounds that exist. Every single organic molecule started out as carbon dioxide snared by ruby P carboxylase. Most organic molecules are digested and respired by aerobic organisms, a process that returns carbon dioxide back to the atmosphere rather quickly. But millions of tons of trees have died and fallen into stagnant swamps where a lack of oxygen prevented decay. The carbon in their wood, all of the cellulose, hemicellulose, and lignin, was converted to coal and did not return to the atmosphere. Petroleum also is probably derived from photosynthetically fixed carbon dioxide. The point is that respiration does not release all carbon back to the atmosphere. Therefore, photosynthesis is gradually causing carbon dioxide concentration in the air to decrease. 3 groups of organisms have had especially important impacts on atmospheric carbon dioxide: coccoliths, mollusks, and humans. Coccoliths are microscopic algae that build shells of calcium carbonate, as do mollusks. When they die, their shells and the carbon they contain sink to the bottom of the ocean and decompose only slowly. All limestone and vast carbonate deposits on the ocean floor represent millions of tons of carbon dioxide removed from the atmosphere by clams, barnacles, and unimaginable numbers of algae. We humans were no different from any other aerobic organism until we made a fateful discovery: how to use fire. Since then, we not only oxidize food in our mitochondria, but we also oxidize wood, coal, oil, and gas, putting carbon dioxide back into the atmosphere and raising its concentration measurably. Why does the concentration of atmospheric carbon dioxide matter? Think of carbon dioxide as a pigment. Its absorption spectrum is low for visible light but high for infrared wavelengths. Visible light from the sun passes easily through the atmosphere. It is not absorbed by nitrogen, oxygen, or carbon dioxide. As it strikes Earth's surface, some is reflected immediately back out into space, and a small amount is absorbed by biological pigments such as chlorophyll in leaves or rhodopsin in eyes, where it powers photosynthesis or vision. However, most visible light has no effect other than to warm rocks, soil, and water, causing them to radiate the extra energy away as long wavelength. Infrared light. Many of these infrared quanta pass directly back through the atmosphere without hitting a carbon dioxide molecule because the concentration of carbon dioxide is so low, but many quanta are absorbed by atmospheric carbon dioxide molecules, causing them to become warmer. This energy is trapped in the Earth atmosphere system and warms our world. This is called the greenhouse effect because the glass in greenhouses works the same way, as does the glass in a parked car. Carbon dioxide is a greenhouse gas. An important balance exists between the atmospheric concentration of carbon dioxide and life. With less carbon dioxide, more heat would be lost and Earth would be frozen, like Mars. With more heat would be trapped and our world would be as hot as Venus, at 800 degrees Celsius, with lakes of molten lead. During the industrial age, we have been adding carbon dioxide to the atmosphere by burning oil, gas, and coal, and we have destroyed forest trees that can remove the carbon dioxide by photosynthesis. The concentration of carbon dioxide is increasing in the atmosphere, and the average temperature is also increasing. This is global warming, and it could cause mean temperatures to be 2 degrees Celsius or 3 degrees Celsius warmer in the next century. Global warming is having numerous consequences. First, surface water of the oceans is becoming warmer, therefore, more water evaporates into the air. Much of our weather in North America comes as winds blow eastward across the North Pacific. The water is cold and the air picks up only enough moisture to keep the Pacific Northwest wet. By the time it moves to the Central Plains states, it has so little moisture left that only grasses, not forests, thrive. But as surface waters of the Pacific become slightly warmer, vastly more moisture will evaporate into the wind and be carried to the Mississippi drainage basin. This increased rainfall could cause much better farming conditions in the Central Plains, and catastrophic flooding in most river valleys where cities are located. El Niño years show the gigantic flooding that results from slight warming in just one area of an ocean. Global warming is also causing rapid melting of snow and glaciers in mountains and of ice caps in the Arctic and Antarctic. It is difficult to comprehend, but Antarctica is a large continent covered by ice 1 to 2 miles thick. As the world's ice fields melt, that water is added to oceans, increasing their volume and causing sea level to rise. Sea levels are rising even more because as ocean water warms, it expands. Coastal cities will be flooded, and so will coastal wetlands where ducks, geese, and hundreds of other species live. The additional freshwater flowing from melting ice caps and flooding rivers has other impacts. Freshwater is lighter, more buoyant than seawater. It spreads outward from a river's mouth as a cap, only gradually mixing with seawater as waves agitate the two. With more flooding, this cap becomes more extensive. Being fresh, marine algae cannot live in this layer, so they are forced into lower, darker waters where they cannot photosynthesize so well. The mixing of freshwater with seawater dilutes the nutrients in seawater, also slowing the growth of algae, and algae are of course the basis of the entire food web in the oceans, all ocean life is ultimately dependent on algae. Another consequence of global warming is that not all areas are being affected equally. Wind patterns are being changed such that certain areas are becoming warmer, others cooler, some wetter, and others drier. This is global climate change. If circulation of ocean currents like the Gulf Stream is altered, climates in vast areas of the world will change dramatically. At what point will we decide that weather patterns have truly changed and that a particular city should be abandoned because of frequent flooding? Some agricultural areas will become too dry to farm, causing at least personal misery as crops fail, perhaps causing massive starvation. New areas will become optimal for farming, but it will take a great deal of confidence to be certain that the new weather patterns are stable enough for people to risk starting again in an unknown area. Millions of lives will be disrupted, some suddenly and catastrophically in floods and others slowly and inexorably as conditions decline. In addition to the problems global climate change causes us, we are also forcing it onto all of the other organisms with which we share the planet and which bear no responsibility for the damage we are doing. Figure B10(1)A. After this tree is killed by the chainsaw, it will stop photosynthesizing and will stop removing carbon dioxide from the atmosphere. Even if converted to lumber and used as part of a house, it will eventually decay or rot or be burned, being oxidized back to carbon dioxide and increasing the amount of CO in the air. The Kyoto Protocol is a treaty designed to reduce production of greenhouse gases. Signed by 166 countries, it went into effect in 1994, but the United States, the largest producer of carbon dioxide, did not sign the treaty. Some people contend that if we do not act decisively, greenhouse gases will build up rapidly. Others point out that China and India, with combined populations of almost 2 billion people, are modernizing so rapidly that soon they will be producing more carbon dioxide than the United States is. They conclude that greenhouse gases will be produced as long as coal and oil are available and people need energy. One human interaction with the atmosphere seems to be going well. The atmosphere protects us from harmful radiation from space by absorbing high-energy cosmic rays, X-rays, and ultraviolet light. Very little of these wavelengths reach ground level. If not for water in the atmosphere, the first two would kill us outright. Ultraviolet light can induce mutations in our DNA and cause skin cancer and blindness, but atmospheric oxygen weakly blocks some ultraviolet light. Fortunately, a byproduct of its absorbing high-energy quanta is the conversion of oxygen to ozone, 3O. 2O, which is even more effective at blocking ultraviolet light. Man-made chlorofluorocarbons from air conditioners and cans of hairspray and deodorant, among other products, had been escaping into the atmosphere and destroying ozone, thus increasing the amount of ultraviolet radiation reaching us. Effort has been made to eliminate production of chlorofluorocarbons worldwide, and its levels in the atmosphere are dropping, thus allowing ozone to accumulate again and continue protecting us. This one success story shows that we have the power to protect the environment that protects us. Figure B10(1)B. Tropical rain forests are not the only forests being damaged. Clear-cutting like this is common in the western United States and Canada. First, chlorophyll, like all other biological pigments, does not use high-energy quanta because they have too much energy. Each is so powerful that it would knock electrons completely away from the pigment, disrupting bonding orbitals and causing the molecule to break apart. Notice in Figure 10.2 that all bonds in the chlorophyll ring system are double bonds that alternate with single bonds. This bond system is excellent for absorbing quanta, but if even a single electron is knocked out, the entire structure becomes useless. It is selectively disadvantageous for a plant to produce a photosynthetic pigment that is destroyed by the light it absorbs. The molecule would break down, and all of the ATP that had been expended earlier in its construction would be wasted. Fortunately for our own molecules, the atmosphere's ozone layer protects us by absorbing high-energy radiation. Long-wavelength radiations, such as infrared and microwaves, have so little energy per quantum that they cannot appreciably boost an electron's energy. Instead, they make the pigment molecule warmer, as in a microwave oven, but this is not especially useful for chemical synthesis. Visible light contains just the right amount of energy per quantum. When one of these quanta is absorbed by the pigment, an electron is activated, raised to an orbital of a higher energy level. We say that the electron and the molecule go from the ground state to an excited state. Under the right conditions, this high-energy, excited electron can be used in chemical reactions. If it is not used, it returns to its original, stable ground orbital by emitting a new quantum of light, one with less energy and a longer wavelength than the one that it absorbed. The release of light by a pigment is called fluorescence. Figure 10.8. If a quantum has the wrong wavelength for a pigment, it passes